Very good evening, everyone. Please confirm if the screen is visible. And is my voice loud and clear? Please confirm. Very good evening, Satyam. I hope you are doing good. Yeah, I'm fine too. Thank you for asking. So, how's the preparation going on? The exam date, CSIR NET Life Sciences exam date is announced. So, I hope you all are preparing well. So, welcome to another session of Brahmastra Series. And today we are going to discuss, we are going to revise the concepts as well as we are going to practice questions from topic molecular cloning and recombinant DNA technology, topic from subunit A of unit 13, that is methods in biology. So as you know, this topic is important from exam point of view because part B questions, you can expect direct part B questions on DNA manipulating and modifying enzymes like alkaline phosphatase, clinofragment of the DNA polymerase, clinofragment, or question on moving nuclease, or a simple question on restriction enzymes. But mainly part C questions are asked where they give you a combination of the restriction enzymes. Like they mention a cloning experiment where they mention that so and so enzyme is used to cut the insert, and so enzyme is used to cut the vector. So which combination of enzyme you will use for cloning? Second type of question, part C question, is usually the figure based, or like they give you a vector diagram, any figure, or restriction digestion pattern, and based on that they can ask you about the number of bands uh, that can be obtained, the size of bands, the size of fragments that can be obtained. So today we are going to discuss questions, part C questions from this topic. So I hope you all are ready. So here comes the first question. I will be displaying the question and giving you some time to solve. So I request everyone to utilize this time because during this 1 hour, you will be solving specific topic based questions. So most of the students, they always email us that, ma'am, we want topic wise questions, right? So here is the practice of topic wise questions. You can utilize this 1 hour and you can practice the questions based on gene cloning and recombinant DNA technology. So very first question is about cloning. You can see the question is you are inserting a gene of 2 kb length. So the size of gene is 2 kb and you are inserting it into a vector of 3 kb to make a fusion protein. So basically you are inserting a gene into a vector. So vector plus gene. So you are making a fusion protein and the gene is being inserted at eco R1 site, and insert has a hind 3 site 500 base pair downstream of first codon. So you can see here a figure is also given where the eco R1 site is mentioned, and it is mentioned that the gene is inserted at the eco R1 site. And within the gene, also you have a hind 3 site 500 base pair downstream of the first codon. So when you are screening for the clone with the correct orientation by restriction digestion of the plasmid, using hind 3 plus BAMH1 and hind 3 plus PST1, the map of the relevant region of the vector is shown below. So this is the vector where the promoter, the GST, terminator, the recognition sites for restriction enzyme is mentioned. Now given below is the pattern following restriction digestion of the plasmid isolated from 4 independent clones A, B, C, and D. Which of the following plasmids show the clone in the correct orientation? So I'm giving you 3 minutes, around 3 minutes to solve this question. Read the question carefully. Although these questions appear to be very difficult, but believe me, these questions, like if you know the strategy of solving such questions, you can easily solve it. Okay. So it's just that you have to be like carefully read the question, pay attention to the details which are given in the question, and accordingly solve. So please try, everyone. No need to hurry. Okay, just take your time and try solving this question. Okay, Satyam, you have already answered. Well done. But I want you to just check your answer once again. Okay, as I told you, right now focus on accuracy. Okay, speed is also important, but accuracy is most important. You have to answer the questions correctly, otherwise you will lose 4 marks, right? So these part C questions are of 4 marks, right? So it's very important that you select the part C questions properly and you solve them accurately. Very good evening, Rinku. Okay, so you have also answered option one. Well, so I can see some more, many are watching the session. So I want everyone, I request everyone to please let me know in the chat your answers. So one hint I can give you that to solve such type of questions, it's better that you redraw the diagram. Okay, based on information which is given, you have to redraw the diagram. Okay. Very good evening, Sarita. Okay, you are saying option C. Please try, everyone. Okay, some more response coming. Very good. Navya, option one. Okay, that's great. See, again and again, I'm saying try to redraw the diagram. Okay, you have one information given that there is a gene and you are inserting it into the vector, right? So gene is there and you are inserting it into the vector. So just this is the this is the vector backbone, you can say, given to you, and here you are inserting your gene. So just redraw this diagram and label all the things. And then, as mentioned in the question, you're performing restriction digestion, Hind 3 plus BAMH 1 and Hind 3 plus PST 1. So now carefully see what size fragments you will get. So on restriction digestion, what size fragments you will get. So I hope you all have understood the question, right? It's a very interesting question. Okay. So most of you have answered option one, but answer option one, that means you say that the restriction digestion pattern given for A clone is correct. So let's check. So first of all, insert size is 2 kb. So 2 kb means that it is around 2000 base pair, right? And then vector size is given 3 kb, that is 3000 base pair. So if you are introducing your gene into the vector, okay, so I'll just use another font color, another ink color. So when you are introducing your gene, okay, so when you are introducing your gene into the vector, the total size becomes what? Insert plus vector. So insert is 2 kb and your vector is 3 kb. So that totals 5 kb, right? So total your gene plus vector should be 5000 base pair, right? Now, this is the information given to us in the question. So here you will just label it because it is mentioned that the gene is introduced in eco R1 site over here, right? So that means on one end you have BAMH1 and over here you will have PST1, right? Then it is mentioned that insert has Hind3 site 500 base pair downstream of first codon. So this is the first codon of the gene. So let's say over here, around over here, we have Hind3 site. The total gene is of how much base pair? Like 2000 base pair. So if this is the region of like Hind3 site from the first codon 500 base pair downstream. Downstream means towards the 3' end. So here, the remaining portion will be how much? 1500, because 1500 base pair, right? Because 1500 plus 500 gives you 2000 base pair, right? The total gene is 2000 base pair, so I'm labeling it like this. And like here, you have the promoter region as mentioned over here, and the terminator region over here, and so on. Now, paying to the important detail that when the restriction digestion, so double digestion means when you are performing the digestion using two enzymes HIND3 and BAMH1. So how many fragments you will get? So this is HIND3. So enzyme will cut over here, right? So again, I'll use the ink color. So the enzyme will cut over here, right? BAMH1 and HIND3. So that means this much portion, this much fragment you will get. So that means one fragment you will get of 500, and remaining this part which is left is how much? The total 5000 base pair. So 5000 base pair minus 500, you should get 4500, right? So that means this should be the size of fragments you should get. Now, if you see at the see all the clones, so on performing the digestion with HIND3 and BAMH1, where you are getting these two size fragments. So here 500 is given, but this is when HIND3 and PST1 digestion is occurring. So that is not correct. We have to check for HIND3 and BAMH1. Okay, so I think this is what the mistake you have done. You have looked for this fragment, look for this digestion. So that is incorrect. So if you see for clone C, 500 fragment is there and 45. So 4 and 5, between 4 and 5. So that means this is 4500 base pair fragment we are getting, right? So this indicates that the answer should be option C. And we can further confirm our answer. So HIND3 and PST1 digestion. So when HIND3 PST1, so the enzyme will digest over here and enzyme will digest over here. So you should get one fragment of 1500, right? And the remaining fragment will be how much? 3500. Vector backbone will be how much? 3500, right? So just see if in C clone this is given. So yes, one fragment we are getting of around 1500, right? This is 1.5, and we are getting 3500. So between 3 and 4. So this means that answer here is option 3, right? That is C clone is correct. So is it clear to you, Satyam, Rinku, Navya, Srinivas? Is it clear how to solve such questions? Interesting, right? So see, the question is not very difficult. It's just that we have to learn the strategy of solving such questions. Okay. The questions appear to be lengthy, but they are not as difficult. Okay. And just an announcement. We are coming up with a Laksh batch for 2025 CSIR NET 2025 and GATE 2026 exam. So if you want to connect with us, if you want to learn the strategy, if you want to prepare for any of these exams or both the exams, you can join our batch, Laksha batch. So for the details, you can call on our toll-free number, or even you can visit stores.biotecnika.org. So along with this batch, you will be getting access to unlimited classes, chat support, study portal, Conceptica series, revision tool, and also test series. Okay, so thank you for confirming, Satyam and Rinku. So now practice such type of questions more, okay? Just to check your conceptual clarity. I will be giving you some more questions now. And also one more thing I would like to tell you, those students who will be with us till the end, you will be getting one freebie with this session, but you have to stay with me till the end, right? So you will be getting some more practice questions along with the explanations and answer based on this topic. The link will be displayed at the end, okay, of this session on your chat. So coming to another question, the CDS of the shortest isoform of human gene A is cloned into a 3.3 kb vector under a CMV promoter at the BAMH1 and XHO1 sites. So CDS is the coding sequence of the gene, okay, of the gene A. It is cloned into a vector. So vector, you can see it is mentioned 3.3 kb. And you are introducing a gene. Now, they have not mentioned the size of the gene. Okay, so this part is the gene. And under a CMV promoters, that is cytomegalovirus promoter at the BAMH1 and XH1 sites. So as the gene is cloned in vector under promoter, so at BAMH1 and XH1 site, so I'm assuming this is BAMH1 site, this is XH1 site, okay. Now, it is mentioned that from the agarose gel and SDS page images shown above, which of the following is most likely true for protein A in HeLa cells? So basically, you can see that the very first figure which is being shown, it is of the this vector being digested with XHO1 and BAMH1. So when you perform the digestion with XHO1 and BAMH1, you can see that two fragments we are getting, right? So this figure is about, which is displaying the restriction digestion pattern of the vector when it is performed with both the enzymes. And second figure is a Western blot. Okay, so Western blotting, although you study in subunit B of unit 13, that is histochemical and immunotechnique. So here you can see it's a combination based question where they have give you restriction, restriction digestion from unit A, subunit A of unit 13, and subunit Western blotting from subunit B of unit 13. Now here it is the Western blot against A on A HeLa cell lysate. So first column basically it indicates the cells transfected with this vector, and second is for untransfected. So you can see some bands you are getting when the when this vector it is being transfected, and when the cell is being transfected, and the second, so HeLa cells when being transfected with this vector, and second is for untransfected, so no bands we are pattern we are seeing here. So you have to analyze these two figures given, and based on that you have to answer the question. So you have to find out which of the following is most likely true for protein A. That is protein A from homo multimers, or it is degraded by lysosome, or it is polyubiquitinated, or it localizes to autophagosomes. So again, I'm giving you some time. Please solve this question. Okay, some of you have already answered. Satyam and Rinku, you are saying option 3. So I'll let you know in some time. Okay, let's everyone, others also solve this question. Pay attention to the details which are given in the question, and specifically the terms you have to pay attention. The details and the terms, like whether you have to choose the correct statement or incorrect statement or true statement or not true statement. Okay, like that. Please solve everyone. I want response from all. You are just here to practice, okay? Nobody is here to judge you. It's just that I want you all to practice, okay? Because then only you will learn. And I hope you all are having a notebook and a pen. Don't try to solve it verbally. As I have already told you, read the question carefully and redraw the diagram. Okay, like you have to sometime based on the information given, you have to assume, and you have to, like, based on the information given, you have to draw the diagram and then understand the concept and then solve. The best part of such question is that you don't have to think out of the box. Whatever information is given to you in the question, accordingly you have to only solve. It's not that you will apply some third concept. No, whatever information is given to you, based on that only you have to solve the question. So analyze both the figures. So when the HeLa cells, they are transfected with this vector. So this is the Western blot showing the bands. The Western blot, I hope you know that Western blot is used for analysis of proteins, right? Southern blot for DNA, Northern blot for RNA, Western blot for proteins. Yes, protein A, correct. So obviously protein will be encoded by gene only, right? So quickly you give me answer, more questions we can discuss. Done, everyone? So definitely for this question, we will consider each option one by one. So first option is protein A forms homomultimers. Now see, these terms, terminology, you need to be you need to be very much clear about what is homo, what is hetero. Okay, these terms need to be very much clear. So homo means same. So like first of all, as I told you, we have to analyze these two figures given. So I have already told you that there is a vector, and in this vector we are introducing a gene. So gene size we don't know, but vector I think it is given 3.3 kb, right? Now, so this is the vector 3.3 kb. Now if you see, and I told you that it is being placed between BAMH1 and XHO1. So when you perform the digestion with BAMH1 and XHO1, you can see that the one fragment we are getting of a little bit more than 3000 base pair. So I'm assuming it is 3.3 kb only, right? And this one fragment we are getting like around 1500, around, right? So that means this is for the gene. So through this we got to know that okay, this is the band for the gene, right? This is the fragment we are getting for the gene, right? And it is around 1500 base pair. And then if we analyze the western blot, the cells transfected with vector are showing multiple bands. Now I told you, like here it is mentioned proteineform homomultimer. Now, if it would have been a homomultimer, we would have not got these different size bands, right? You can see they are in the range of 90 to 53 kilodalton. So if they would have been same, like we would have got single band, right? So it doesn't indicate homomultimers, right? So therefore, uh, first option cannot be the answer. Then protein is degraded by lysosome. So usually lysosome, if a protein is degraded by lysosome, a smear band of low molecular weight is indicative of degradation. So yes, we are getting like around 9 kilodalton. You can see a smear we are getting, but it results into broad smear and also not specific bands in the higher molecular weight range. So that is why this option is also omitted out. Now, protein is polyubiquitinated. Polyubiquitinated is a Ubiquitination is a form of post-translational modification where a ubiquitin is being attached to a amino acid residue, particularly lysine. So yes, through studies we know that polyubiquitination leads to smear due to addition of multiple ubiquitin molecules. So if we are getting these bands in this range of 53 to 90 kilodalton, and also the smear band of low molecular weight, it aligned with this scenario. So yes, this could be the answer. And last is protein A localizes to autophagosome. So we need some more additional experimental evidences for this. So that is why this cannot be the answer. So very good, Satyam and Rinku. Your answer is absolutely right. The answer is option 3. So just see, just to answer this question, you need to be familiar with the concept of restriction digestion. You need to be cloning. You need to be familiar with the concept of Western blotting. You need to be familiar with the terms like homomultimers, hetero, polyubiquitination, like about post-translation modification. Also, you need to be familiar with, right? And even like what is the function of lysosomes, autophagosomes. Now coming to another question. Which one of the following intermediate enzymatic reactions would be most effective in facilitating ligation of blunt-ended insert fragment and a vector digested with eco-R1 restriction enzyme? So this question is based on several DNA manipulating and modifying enzymes. And you can see that in the options, moving nuclease is given, shrimp alkaline phosphatase is given, cleano DNA polymerase is given, polynucleotide kinase is given. So definitely to answer this question, you should be familiar with the function of these enzymes, right? So go through this question and tell me what do you think is the correct answer here. Pay attention to the detail, like insert, whether it is blunt ended or it has a cohesive ends. What is the ends of the vector? What is the concept of joining insert with a vector? What kind of ends you can join together, right? While creating a recombinant DNA molecule or while joining two DNA molecules. So think about it and answer this question. Option 4. Okay, Satyam and Rinku are saying option 4. Fine. So I'll just confirm your answer in a minute. What about others? I need response from everyone. Please, this hesitation has to be removed. Okay, if you hesitate in answering, then you will not get confidence, right? See, when you try, when you solve the question, maybe first time when you're solving, you didn't get it correct, right? But this is how we improve, right? This is how we learn. And you will see that by the time, like when you solve more and more questions, revise the concepts, you will see improvement in your question solving. And definitely more the questions you answer correctly, more you get confidence, right? So for CSI NET Life Science exam, um, developing confidence is also very much crucial, because if you're confident enough about your preparation, about your question solving, definitely nobody can stop you from cracking the CSI NET Life Science exam, right? Okay, Satyam, you mentioned the treatment of insert with cleano DNA polymerase followed by treatment with shrimp alkaline phosphatase. So Clino DNA polymerase is used to fill in sticky ends. Yeah, you are right about that. But pay attention. So you have mentioned very good. I'm glad. I really appreciate your answer, Satyam. You know the concept that okay, Clino DNA polymerase, Clino fragment we use to fill in the gap, and like we can convert the sticky end into blunt end. But the point is, insert is already blunt ended. Did you pay attention to that? Did you pay attention to this detail? Your insert is already blunt ended. So what you will do by treating it with clino DNA polymerase, right? So that is why I told you, I told everybody that please pay attention to the details given in the question. I hope it is clear to you, Satyam, what I'm pointing out. Insert is already blunt ended. Now, if you pay attention, vector is digested with EcoR1. Now, EcoR1 generates sticky ends. Even the recognition site for this enzyme is given, GAATTC, and you can see that they have given over here. This slash means that okay, enzyme cuts over here. So, blunt-ended fragments are generated when the enzyme cuts just in middle of recognition site and at the same position on both the strands, which is not the case over here, right? So even in option 2 also it is given insert with clino DNA polymerase. So simply if you pay attention to this detail, you can easily omit two options out of four that okay, option 2 and 4 cannot be the answer, right? So definitely you should know the concept. What is the function of moving nuclease? What is the function of shrimp alkaline phosphatase, clino DNA polymerase, polynucleotide kinase, right? But even you have to pay attention to the details. That is most important that is given in the question. So, what is the role of moving nuclease? It is specific, it's an endonuclease and it specifically cuts the single stranded DNA or RNA. And if whenever there is any extension, single stranded extension, it cuts. Okay, so single stranded extension and double stranded DNA cuts. So it's actually single stranded specific endonucleases. That is, wherever it sees a single stranded extension in double stranded DNA, it cuts there. Like here it is shown. This is the single stranded portion, right? It doesn't have a complementary strand. So moving nucleus can cut it and convert your sticky ends into the blunt ends, right? Because this is an overhang region, right? Some stretch of sequence is not a single stranded of double stranded DNA, so it is converted to blunt end. Then clino DNA polymerase, so actually Escherichia coli DNA polymerase 1, when you treat with a mild protease, it results into formation of two fragments. The larger fragment is called as clino fragment, and this clino fragment, it has the polymerase activity, it retains the polymerase activity, and also have 3' to 5' exonuclease activity. But it lacks the 5' to 3' exonuclease activity of Escherichia coli DNA polymerase 1. So actually, when you perform when you treat the Escherichia coli DNA polymerase 1 with a mild protease, you get two fragments. One is larger fragment called as clino fragment and the smaller fragment. So smaller fragment only has a 5' to 3' exonuclease activity. So clino DNA fragment, it fill in the nick, like whenever any single stranded region is there, it adds the nucleotides over here, right? But because it lacks the 5 prime to 3 prime exonuclease activity. The remaining existing nucleotides, they are not being replaced. Then coming to the role of shrimp alkaline phosphatase. So it's a phosphatase enzyme. That means it will remove phosphate group. And polynucleotide kinase opposite, just work opposite to that of alkaline phosphatase. It adds the phosphate group, and phosphate group is added at a 5 prime end. So you can see alkaline phosphatase remove the phosphate group. And why alkaline? Because it works in alkaline pH. That is why the name is alkaline phosphatase given. And polynucleotide kinase, it removes the phosphate from the polynucleotide kinase work opposite to that of alkaline phosphatase. It adds the phosphate group. So here you can see initially phosphate group is not there. So this is your 5 prime to 3 prime, and this is 3 prime to 5 prime. So DNA is double stranded and like two strands. So one is 5 prime to 3 prime, another complementary is 3 prime to 5 prime. So you can see at 5 prime and phosphate group is added by polynucleotide kinase. Now it is mentioned that insert is blunt ended. So you don't have to convert your insert into blunt ended. It is already blunt ended. But your vector is digested with eco R1. So definitely it will have a sticky end. Now we know this concept that you cannot join a sticky and that is a DNA molecule with sticky end to a blunt end molecule. Blunt end molecule can be connected to blunt end with the help of DNA ligase, or sticky end connected to a sticky end through complementary base pairing. But you cannot ligate a blunt end DNA to the sticky end vector. So that means either both the insert and vector have to be blunt ended or both have to be sticky ended. So here, as the vector is digested, right, as the vector has a overhang region, it is digested with eco R1. So we can convert it into the blunt end, right? It will be easy. So which enzyme we can use? Moving nuclease we can use, right? So vector with moving nuclease, it is given in option 1. So yes, it sounds correct, right? And in option 2, I told you insert with DNA polymerase. Now insert is already blunt ended. What will you convert? Sticky end is converted to blunt end. DNA polymerase can convert sticky end into blunt end, but it is already blunt ended. So 2 cannot be the answer. Similarly, 4 cannot be the answer. And here treatment of vector with polynucleotide kinase and then insert with move being nuclease. So here we are, so again, insert is already like blunt ended. So the answer here is option 1, right? Because overhangs in the vector after eco R1 digestion can be removed by nuclease, and then we can add it with the, like alkaline phosphatase can remove the phosphate to prevent religation of the vector. And DNA polymerase, this point I have already told you, could also be used to convert sticky end into blunt end, but it has to work on the vector, not the insert. But in the question, insert is being treated with DNA polymerase, which is not that. So yes, answer will be option 1, Sarita. Very good. Rinku, it will be option 1. Satyam, it will be option 1. I hope it is clear to you all now, right? So not very difficult. Now, let's uh, discuss a statement-based question now. A researcher attempted to clone two genes, X and Y, independently in a plasmid vector. Okay, independently. Like first, the researcher attempted to clone X, then again, like to the vector gene Y. So, a researcher attempted to clone two genes, X and Y, independently in vector for overexpression and purification in Escherichia coli. So, all attempts to clone gene X were unsuccessful. This is important information. Whereas gene Y could be cloned easily. That means successful. Now, when the researcher attempted to clone gene X in the plasmid clone containing gene Y, gene X could be cloned. So, initially, the researcher was trying to clone the genes independently. So, X and Y. So, this was unsuccessful. This was successful. And now when it tried to clone both the genes together, like gene X along with gene Y, right? In the plasmid clone containing gene Y, the experiment was successful. The X could be cloned. So following statements were proposed to explain the above results. A is protein encoded by gene Y is not lethal to the cell. B, gene X has introns which prevents its expression in E. coli. C, expression of X protein is lethal to the cell. D, Y gene product inhibits activity of X protein. So, which one of the following options represents a combination of correct statement? Very important term. Correct statements to explain the observations. So, go through this question, read it carefully, and answer it. What do you think is the answer here? Okay, Satyam, you have answered option C. Uh, only A. Option 3, okay. What about others? Consider one statement at a time. Okay. I want response from everyone. So when attempting to clone gene X only, the result was not successful. When attempting to clone the gene Y independently, successful. And when attempting to clone gene X in plasmid, clone containing gene Y, X could be cloned. These are the three things that you have to keep in mind. Okay, Satyam, you are saying option 4. Okay. Yes, please try everyone. One more minute I'm giving. So do you all need some more practice questions? Yes? Yes, are you all interested in practicing more questions? Other than the questions we are practicing here, do you need some more questions to practice? Yes or no? Please let me know in the chat. Okay, Phyllis, you are asking, could you please recommend any good book for understanding the basics so that I could build a strong base for genetic engineering apart from T.A. Brown? Phyllis, well, I will suggest that to like there are certain things which are not given in the books as well. So you can always connect with us if you are not connected. Like you can always join us. And apart from T.A. Brown, there are some more good books. Primrose is also there, which you can refer. So this is one book that I can tell you right now. See, actually, the point is reference books we can refer, but the point is like there is a lot of details and informations which are given, and it, like although they are in English, but again it depends. Like someone finds T.A. Brown much better than the Primrose or other book. So it all depends on person to person. Okay, like sometimes some book diagrams, some finds easy and easy to grasp in comparison to other books. So you can refer 2 3 books, and it's up to you, whichever you find comfortable, you are comfortable with, and the language, the way the content is written, you find easy, that you can refer. Okay, the books are all good. These standard reference books are all good. Okay, so coming back to the question, so the information I have already told you. So the very first thing is, so I have already told you that when the two genes were cloned independently, they were for X, the experiment was not successful, but for Y it was successful. Then when the researcher attempted to clone gene X in the plasmid clone containing gene Y, it could be cloned. So statement A is protein encoded by gene Y is not lethal to the cell. So this is correct, right? Because when you are trying to clone gene Y, it can be cloned easily. So that is why it is correct. Lethal means it results into killing of the cell, which is not happening here. Then statement B is gene X has introns which prevents its expression in E. coli. If this would have been the case, then when we are trying to clone it along with gene Y, then also it would have been unsuccessful. Then also the cloning would have been unsuccessful, but we are not getting the result here. So this is incorrect. Then statement C is expression of X protein is lethal to the cell. Yes, we can say this because when we are trying to clone X independently, the cells are like, we are not, the result is unsuccessful, right? So this is only possible when expression of X protein is lethal to the cell. So this is correct. Statement C is correct. And statement D is Y gene product inhibits the activity of X protein. So yes, this is also correct because because this is like, then only gene X can be cloned, right? And it is mentioned that when we are trying to clone it along with the A, along with the Y, then yes, X could be cloned. So this is only possible when Y gene product inhibits activity of X, that is lethal protein. So statement D is also correct. And as we have to choose the combination of correct statements, so answer here is A, C, and D. So wherever you have B option given, you can easily omit 2 options out of 4 if you are following this strategy. Now you're left with only 3 and 4. So when you say C is correct, that means answer is option 4. And even we have confirmed the D part also, the D statement also. Okay. Okay. Now, these type of questions are frequently asked. This one particularly frequently asked. Given below are names and recognition sequences of a few restriction enzymes that are used for cloning experiments. The cleavage site of each enzyme is indicated by the asterisk sign. So you can see over here, like all the recognition site for restriction enzyme EcoR1, HINC2, EcoR5, VAMH1, BG3 is given. BGL3, BGL, yeah, BG3 is given. So given below are different vectors. Column A. Column B is insert fragment generated by digestion using above enzyme. Which one of the following options represents the correct combinations of the vector and insert respectively? See, these terms are very important, whether correct statement is asked or incorrect is asked. Okay. Sometimes they mention one correct to incorrect statement or vice versa. So you have to choose the option which represents the correct combinations of the vector and insert, which generate compatible ends for ligation. To answer this question now, welcome, fellas. So pay attention to the recognition sites of restriction enzyme. You can see that some of the enzymes, like if you see HINC2 and if you see EcoR5, they generate blunt ends, right? Whereas if you see BAMH1, BG3, EcoR1, it generates sticky ends. So that concept I told you, right? Blunt end can be joined to a blunt end, sticky end to a sticky end, right? So that concept you have to remember here. Now, if you are getting confused how to read this table, then definitely it requires practice. Okay, I will be telling you a strategy how for such type of questions you should read the table. Okay. Eco R5 with 1. So none of the option A 1 is given, Satyam. Just check the options also. Nowhere A1 combination is given. Either A2 or A5. You have to look at the options also. Rinku, you are saying option 1. Okay. A2, B5. So Rinku, B, that is eco R5 generate blunt end, and you are saying 5, that is BG3, so it generates sticky end. Is this combination possible? That vector and insert you cut with the enzyme, which one generates sticky end and another generate blunt end. That is not possible, right? See, always remember, either we use the same enzyme to cut the vector and the insert, or we use a combination of enzyme which can generate the compatible end, right? So here the combination is given in such a way that you have to look for which enzymes can generate the compatible end. So basically, first you need to understand about what are restriction enzymes. So we know that restriction enzymes, these are the endonuclease enzyme, that is they cut within the molecule. Endo means within. So they cut within the DNA molecule. And the speciality about these restriction enzymes is that they don't cut randomly. They recognize a specific sequence and they cut at that. They recognize a particular sequence and cut at a specific site. So that is why restriction enzymes, because they are used to cut the DNA molecule, we call them as molecular scissors. And when the enzymes cut, they can generate two type of ends. So here I'm taking an example of ALU1 enzyme. So you can see ALU1 recognizes AGCT, 4 base pair recognition sequence it has. N can be any nucleotide. Now, when the enzyme cuts in the middle of the recognition sequence at the same position on both the strands, the ends generated are blunt ends. On the other hand, if you take the example of EcoR1, which recognizes 6 base pair sequence GAATTC, so this is double stranded DNA shown here. So this enzyme, it cuts on one strand at this place, and on another strand, it doesn't cut at the same place, it cuts 3 to 4 nucleotide away from it, that is over here. So because of this type of digestion, the ends generated are cohesive or sticky end. So you can see there's a overhang region, a single stranded region, which is single stranded region is present, overhang region is present. And why we call them as sticky end? Because they can stick back by transient base pairing. So that is why they are sticky or cohesive ends. Now, to answer this question, we have to, as I have already told you, that you have to use the vectors and insert fragments for cloning that have compatible ends. So we will consider, like, you have to pay attention to which all enzyme generate blunt ends. So I have already told you EcoR5 and HING2, they generate blunt ends. So anywhere EcoR5 coming with HING2, that can be used. That is B2. If you see in option 2, B2 is given. So we can look for it. So yes, this combination is possible. Because blunt end, if to insert in vector having blunt end, simply they will be joined by DNA ligase enzyme. Ligase enzyme acts as a glue and join, connect a phosphor, make a phosphodiester bond and connect these two DNA molecules. So, and if you see A1, so I told you A1 is not possible, right? Because A is EcoR1 which generates sticky end and your EcoR5 is generating blunt end. So this combination is not possible, right? So A1 is not possible, but B2 is possible. Now you can see B2 is given only in option 2. In option 1, it is B5, so this is incorrect. And in 3 and 4, B is not given. So either you can take one more to cross check your answer. So like we can take D, okay, because D is given here also and here also. So whether it is D1 or D4. So first I'll take D4. So BG3 with BAMH1. So BG3 with BAMH1. Now kindly note the sequences which are given here are like of one strand only, 5' to 3'. They are written 5' to 3'. So if this is 5' to 3', this is also 5' to 3'. So I have to consider 3' to 5' strand of another enzyme. Okay, so if I'm considering BG3 5' to 3', so BAMH1 sequence is this. I'll write it here. GGATCC. So this is 5' to 3'. What will be the 3' to 5' sequence? C, C, T, A, G, G, right? Now I'll compare 3' to 5' sequence. Like I will see whether complementarity is there or not. So I will write here BG T sequence GAT CT. Okay, so this is 5' to 3', and enzyme cuts over here, right? So this is of BG3, and now I'm writing the sequence C, C, T, A, G, G, and this is 5. So enzyme cuts over here. So you can see at least these sequences are complementary. G pairs with C, A pairs with T, T pairs with E, C pairs with G. So yes, this combination can be used. They can stick back, they can join, they have the complementarity. So D4 seems to be correct. Now when D4 is correct, so definitely D1 is given here. It cannot like this combination B2, D4. So E1 also you can cross check. Now last we are left with E1. So E1 is HINC2 and EQR5. So HINC2 and EQR5. So again, they can be used, right? E1. Okay, so E1 is possible. So B2 is right, D4 is possible, E1 is possible. So that means your answer is option 2. Sarita, your answer is correct. Satyam and Rinku, is it clear? Answer will not be option 1 or 4, it will be option 2. So when you have so much combinations given, you can just you just pay attention to one combination and check whether it is correct or not, then move over to next, right? So we have basically omitted options. We know that okay, B2 is right, so one like combinations, correct combinations we have to choose. So you can cross, like you can then next take D4 and then you can next take E1. The answer here is option 2. Is it clear to all? Sarita, complementarity, I'm saying that we have to like see one strand if 5. By default, the sequences written are 5' to 3'. So whatever sequences you see here, they are all written 5' to 3'. Okay. Now you should, we know very well that DNA is double stranded. So although the complementary, although the 3' to 5' sequence is not given, but we have to write it when we are checking for complementarity. So I am checking here complementarity between the BG3 and BAMH1. Okay, BG3 and BAMH1, that is my D4 combination. So what I have done is I have written both the strand sequence of BAMH1. So GGATCC is already given. So I have written the complementary strand sequence, 3' to 5' sequence. Now, obviously, we know that if 5' to 3' of one sequence can bind with 3' to 5' of another, right? They need complementarity. So I am considering BG3 5' to 3' strand and I am comparing it with the BAMH1 3' to 5' strand. So this is what I have written here, and I have told you that yes, G pairs with C, A pairs with T, T pairs with A, C pairs with G. So they have complementarity, and this combination is possible if, like, if you perform the digestion of insert and vector with combination of using these two enzymes. Clear to you, Sarita? So last question for today. Given below are the approximate lengths of DNA fragments obtained on agrogel electrophoresis following restriction digestion of a 3 kb circular plasmid with different restriction enzymes. And they have given you like they have given you that when you perform digestion with BAMH1, you will get 2 fragment. When you perform digestion with HINC2, you will get 1 fragment, and so on. Now, based on the above information, which of the following statement is incorrect? See, this is very important term. Here you have to choose the incorrect statement. So again, to solve this question, you have to go through each statement one by one and you have to solve it. Now clear, Sarita? That's good. Okay. Thank you for confirming. So that is what, we have to learn this thing that certain things we have to assume also. Like, okay, one strand information is given 5' to 3'. So automatically to check the complementarity, you have to write the 3' to 5' sequence also of the same strand, of the strand given. Okay. Sequence given, accordingly you have to write the complementary sequence. Yes, solve this question now quickly. Last 5 minutes. So redraw the diagram based on, like you have to draw the diagram based on the information given in the question, because then only you can answer this question correctly. It all comes with practice. Okay, you have to read the question carefully, practice drawing the diagram, vector, insert diagram according to the information given in the question. So did you learn some new concepts today? Can I get some thumbs up? These concepts you will not find in books, right? Basic information it is. Okay, thank you for confirming, Phyllis. I hope the same from others. Yeah, that's great. Thank you. So first of all, I must really appreciate you all, especially Sarita, Satyam, Rinku, Phyllis. You have been consistent in answering the questions. It doesn't matter whether you are answering it correctly or incorrectly at this stage. The most important thing is at least you are trying, and that is the good part. Because when you try, then only you'll understand how much like you can analyze your preparation and your performance, right? So you still have time. You can work on your concepts, and also you can work on question solving, right? So work on accuracy and speed for question solving. Yes, quickly give me the answer for this question. So you have the approximate length of DNA fragments obtained on agarose gel electrophoresis. So when you perform the restriction digestion of 3 kb circular plasmid with a different restriction enzyme. The total size of the plasmid, and you can see it is circular given, not linear. Now here is one more concept which I would like to tell you here. I don't know whether you know this or not. To determine the number of fragments, there is a very easy rule that you can remember. Whenever we talk about circular DNA, so always remember the number of restriction sites, the number of fragments obtained will be equal to number of restriction sites present. Like for example, if you see here, we have 4 sites for enzyme Hind3. So number of fragments obtained will also be 4. Okay, like enzyme will cut from here and here, so 1 fragment. Then enzyme cut from here and over here, so second fragment. Then over here, so third fragment, and this is the fourth fragment. But for linear DNA, there is an n+1 rule. N+1 rule where n is number of restriction sites. So if 4 restriction sites are there, the number of fragments obtained will be 5. 4 plus 1, 5. So like over here, enzyme cuts. So this is 1 fragment you get, right? Then enzyme cuts over here. This is another fragment you get. This third fragment, here to here, fourth fragment. So these arrows indicate restriction site, okay, for enzyme Hind3. And this is the fifth fragment. So in the question, the information given to us is that there is a 3 kb vector. So I can draw the circular plasmid. And the information given is, like you can see over here, okay. BAMH1 gives 2 fragments. So I told you for circular plasmid, the number of fragments is equal to number of restriction sites. So this indicates BAMH1 has 2 restriction sites for 2 restriction sites for BAMH1. For HINC2 and EcoR1, one fragment, so that means one restriction site is present. And even you can see 2.5 plus 0.5 gives you 3 kb only. Now, when you perform double digestion, that is when you're performing digestion with two enzymes, EcoR1 and BAMH1, you are getting 3 fragments. EcoR1 and HINC2, 2 fragments, and BAMH1 plus HINC2, 3 fragments. So, based on these easy information, we will draw the diagram. Okay, so you can see that BAMH1, 2 sites are there. HINC3 and EcoR1, one site is there. And if you pay attention to over here, that BAMH1 and HINC2, when you perform the digestion, you get 2.5, which you are getting over here also, and 0.2 and 0.3. So 0.2 plus 0.3 gives you 0.5. So that means HINC2 site is present between two BAMH1 sites, because then only 0.5 can be broken into 0.2 and 0.3. So that is why I have drawn the diagram where BAMH1 site, in between you have HINC2, and EcoR1 will be somewhere else. So which we can determine based on EcoR1 and like other restriction digestion patterns. So first is HINC2 and EcoR1, they have a single recognition site each in the plasmid. So is this correct? Yes, it is correct, right? HINC2, just now I told you according to the rules. So one fragment we are getting, that means one site. So this is correct. Statement 2, HINC2 is located between two BAMH1 sites. This is also, just now I have told you, right? By seeing the digestion of BAMH1 and HINC2, since we are getting 0.2 and 0.3, this indicates that site is between the two BAMH1 sites. So statement 2 is also correct. So this is what I exactly the same what I'm saying, whatever I'm speaking, I have written here. And then you have statement 3, that distance between EcoR1 and BAMH1 site is less than between HINC2 and BAMH1. This is incorrect, right? HINC2 and BAMH1 is only 0.2 or 0.3. But if you see, EcoR1 definitely will be far, right? So it will be more, because you can see when we are performing digestion with EcoR1 and HINC2, we are getting 1.3 kb and 1.7 kb. Or EcoR1 and BAMH1, we are getting 1 kb, 1.5 and 0.5, right? So like EcoR1 must be here, because then only when you perform digestion with HINC3 and EcoR1, you get 1.5 plus 0.2, that is 1.7. And here 1 kb this must be. So that is why this totals 1.3 kb, right? So this statement is incorrect, and I hope you remember we have to choose the incorrect statement. So answer here must be option 3. We check for option 4 also, that HINC2 is located close to one BAMH site than the other. This is correct. You can see this is this HINC2 is located closer to this BAMH1 than this BAMH1, right? So 0.2 is smaller than 0.3. So statement 4 is correct. And since we have to choose the incorrect statement, so answer here will be, so we have to see the options. So options here is like we have to choose the incorrect statement, right? So it will be option 3. That just a minute, what I have told you. According to this option 3. Okay. Is it clear to everyone? So I hope you have enjoyed the session today, and all the very best. Take care. And the link you will get on the chat. Okay, just check your chat. The link for the PDF for the some practice questions is uploaded. Okay, so you can check on the chat. There is a link for part C questions, more part C questions based on this topic practice. Okay, so thank you everyone. Take care. We'll see you in another session.